Hey you guys. Okay, I know that you guys did go over Candida albicans and rhizoids and germ tubes, but I do want to go over them again with you, also discussing how to set up the lab for that, how to set up a germ tube test. I just want you to know it's the 4th of July and I'm sitting outside on my deck and the neighbors are setting off fireworks. So if you hear any explosions, nobody's shooting at me for doing this. This is just everybody celebrating our fabulous 4th, okay? So do not be alarmed. Our neighbors love their fireworks. Okay, so we're going to talk about Candida albicans, germ tubes, and rhizoids. Candida albicans is actually considered a yeast in the lab, but it is actually dimorphic. It'll bud like a yeast, but it'll also create germ tubes and pseudohyphae. And as you learned, those dimorphic organisms are always the worst as far as human infection goes because you treat one part and then if it gets stressed or whatever, it'll turn into something else, and then it's harder to deal with. So yeah, you obviously can hear the fireworks. So two Candida species actually grow germ tubes, Candida albicans and Candida dublinensis. Okay, but for the most part, when you see germ tubes, it's going to be Candida albicans because it's far more prevalent of an infection than Candida dublinensis. Candida albicans may grow rhizoids on agar, but not always. Um, if you see rhizoids, it's a good indication it's probably Candida albicans. But if it's an oval budding yeast and it does not have rhizoids, that doesn't mean it's not Candida albicans. And you will see Candida albicans far more than any other kind of yeast or fungal infection. Okay. Germ tubes and the secretion of proteinases are virulence factors for Candida albicans. Excuse me. So here's what the rhizoids. Sorry, I just totally burped. You did. I'm sorry. Okay. I'm just passing that off as that was another firework. So the rhizoids look like this on a plate. They don't usually create rhizoids on a MAC, but on blood and CNA for sure, and chocolate as well. So here you'll see them. They look like feet. So if you hear somebody say, "Oh, they have feet," that means they're growing rhizoids. Okay, so now we're going to talk about germ tubes because rhizoids are pretty self-explanatory. Germ tubes add to the virulence of Candida albicans, and I wanted to show you why because I thought this was really interesting, and I just learned this today, so I thought I'd share. These are liver cells up here. Okay, and these are Candida albicans growing germ tubes, and these germ tubes also secrete this proteinase, so it will break down the protein in the liver cells here, shown here. So you can see how they're really infiltrating these liver cells here. Now here is a Candida albicans that is not, or I don't know if it's Candida albicans or some other yeast that is not producing germ tubes. And you can see the clear delineation of there's the infection and they're not really infiltrating these at all. And the exact same with this. These have germ tubes and pseudohyphae, and you can see them really getting in there, whereas here you can see there's almost like a barrier. They can't go any further. They're trying, but they can't infiltrate as much as that. Oops. Stupid PC. Okay, so identifying germ tubes, here are the lab instructions. First off, I want to say 95 to 97% of Candida albicans will grow germ tubes or develop germ tubes in the broth, reagent broth. Germ tube broth usually consists of fetal bovine serum. The reagent usually comes in a powder and has to be reconstituted. We use deionized water, and then we aliquot a half a milliliter of that into test tubes, and then we freeze them. Since we don't do many germ tubes, we freeze them just to be able to keep them longer. And then when we need one, we just take one out of the freezer and thaw it. Every day you do a germ tube, you have to run a positive and negative control, which is called each day of use QC. And you have to obviously use known organisms there, so you can guarantee that what your patient result is will be consistent with the positive and negative that you should see when you run your QC. So the positive control is obviously Candida albicans, and the negative control, we use Candida tropicalis. Candida tropicalis actually looks like round clusters on a wet prep. I'm going to try and get some germ tube media for lab, but I'm not sure if I can. So here's the lab instructions. You take a loop full of your isolated colony and you emulsify it into the gram tube media, in the germ tube media, in the test tube. It's all liquid, so you kind of mix it in there. And then you cover the tube gently with a lid. If you put the lid on tight, as the broth heats up in the incubator, the lid will pop off and shoot off somewhere in your incubator, and then you won't be able to find it. So make sure you just cover it lightly. Okay, and then you incubate it in an air incubator for 2 to 4 hours. After it's done incubating, you shake the tube very gently just to suspend the organism in the broth because it has a tendency to sink to the bottom since has weight, you know. And then you pipette 1 drop onto a slide and you cover it with a cover slip and you observe it under a microscope at 40x, no oil, remember 40x, with the condenser down. Okay, so here are germ tube positives. The thing that you should remember is germ tubes, they look like spoons. There's no stricture here. It just looks like a spoon. Okay, germ tube negative, there's this stricture. It looks like somebody's like strangling them with a piano wire or something. Not that I read a whole lot of true life crime or anything like that. But anyway, when you see a stricture at the neck, you know that it's germ tube negative. If it looks like a spoon, it's germ tube positive. So pass for class is spoon. Okay, pass for class is spoon. There's only one part to this, so. Okay, so here's nice. This is a good thing to see. So here's the mother cell, and you can see now this is budding. This is not a germ tube. This is a budding cell, okay? Here, however, see how this looks like a spoon. There's no stricture. It doesn't look like anybody strangling it. This is a germ tube. This is the virulence factor that kind of sneaks its way into the cells, secretes that proteinase that will like break down the proteins in the tissue cells and will allow it to infiltrate easier. Okay, this is not a germ tube. This is not a germ tube. These are all budding yeast. This is a germ tube. Okay, so I want to ask you now, are these germ tube positive or are they germ tube negative? These are germ tube negative. You can see that there's strictures here, right? They don't look like spoons. I'm not sure what they look like. This looks like a strangled spoon, but it's not a spoon, okay? So you see how there's strictures. This one, what do you think? This is kind of the exact same one we saw right before. These are budding cells. This is the germ tube. Okay, so I just wanted to kind of give you guys a review because these are things like this germ tube, you're going to be doing these in the lab. So I wanted to make sure that you understood how it works, how to do the test, and what to look for results. Okay, all right. Thanks, you guys. Have a good day. Happy 4th. Well, by the time you get this, it's going to be way past the 4th. So, okay, never mind. See ya.